In the 1980s, Bob Horwitz and co-workers at MIT, Cambridge, Massachusetts started to do forward genetic screens for C. elegans mutants with defects in the invariant pattern of cell death. What were they looking for, and what did they use as an assay? First, what were they looking for? They thought that if there are genes that are required for the 131 cells to die, then it should be possible to find animals homozygous for loss of function mutations in these genes by looking for mutants in which at least some of these 131 cells no longer died. And that's what they were looking for. What did they use as an assay? Of the 131 cells that die during C. elegans development, 113 die during embryogenesis. However, when you look at an embryo by DIC microscopy, you only see a handful of these apoptotic corpses, even if you scan the entire embryo. And that's because, as I mentioned earlier, in C. elegans, cells die very quickly and the cell corpses are swiftly engulfed and degraded. They don't persist. And that's why at any given time during embryonic development, you only see a few corpses. The small number of corpses you see make it hard to look for mutants in which some of these cells no longer die. To look for fewer corpses in embryos by DIC microscopy was therefore not a good assay. Fortunately, a few years earlier, in 1983, Hedgecock, Solston, and Thompson at the MRC LMB in Cambridge, UK, had identified two recessive mutations that cause a block in the engulfment of the 131 cells that die. They called the phenotype that these two mutations cause cell death abnormal, or SED phenotype. And they named the genes that these two mutations define SED1 and SED2, the first and second gene identified in C. elegans that can mutate to cause a cell death abnormal phenotype. Just a note, at the time, the genes SED1 and SED2 were defined only by these mutations. Where in the C. elegans genome they were located and what their gene products were was unknown. It took years before, using genetic mapping and rescue experiments, these two genes were cloned and their identities determined. And this is true for the mutations in genes that we will talk about today and in the next couple of sections as well. In SED1 and SED2 mutants, apoptotic corpses are not engulfed. These two genes are therefore required for one aspect of the last step in apoptosis, the engulfment of the apoptotic corpses. What happens to these unengulfed corpses? They persist for a long time. And this can easily be seen in L1 larvae by DIC microscopy. In wild-type L1 larvae, there are no apoptotic corpses visible in the anterior region or head region of the animal. In contrast, in SED1 or SED2 mutant L1 larvae, there are 20 to 30 apoptotic corpses in this region. It's really easy to see, and there are lots of those corpses. This is a great assay. It allows you to screen for mutants in which there are fewer of these corpses. For this reason, rather than mutagenizing wild-type hermaphrodites, Horwitz and co-workers mutagenized SED1 mutant hermaphrodites. And they used hermaphrodites homozygous for the SED1 allele E1735 in particular. They allowed the P0s to reproduce as well as their F1 generation. The resulting F2 generation, they then screened as L1 larvae, and they screened for L1 larvae that had fewer apoptotic corpses in the head region. They screened the F2 generation of a total of 4,000 F1 animals. If there's a gene which when mutated can block the death of the 131 cells, then the progeny of 4,000 F1 animals should be enough to identify at least a couple of loss of function mutations of this gene. Hilary Ellis, who performed the screen, identified two independent F2 animals, basically two F2 animals from different clones, that had no apoptotic corpses in their head regions in the L1 stage. The two mutations that she had identified were named N717 and N718 because they were the 717th and 718th mutation identified in the Horvitz lab at MIT. Both mutations turned out to be recessive, which means that the disappearance of the apoptotic corpses in the head of SED1 mutant L1 larvae was only observed in animals that were homozygous for either N717 or N718. N717 and N718 therefore most likely represent loss of function mutations of some unknown genes. Because both mutations behaved the same in all the analyses that Hilary Ellis subsequently did, I will therefore for now just focus on N717. What could cause the phenotype observed in the SED1 E1735 N717 double mutants? Again, SED1 E1735 causes persistent apoptotic corpses in the L1 stage. In the double mutants, persistent corpses are no longer present. There are at least 3 different scenarios that could explain this phenotype. Think about it for a couple of minutes. First, in the mutants identified, engulfment might take place again, which means that the apoptotic corpses are engulfed during embryogenesis and no longer persist in L1 larvae. Second, in the mutants identified, the 131 cells that normally die might not be generated. And third, in the mutants identified, the 131 cells that normally die might be generated. However, they might fail to die and instead inappropriately survive. To determine whether cells are engulfed again, Hilary Ellis looked at SED1 E1735 N717 embryos by differential interference contrast microscopy. And what she didn't find were apoptotic corpses. For this reason, she concluded that N717 does not reactivate the engulfment process. Otherwise, she would have seen apoptotic corpses in the embryos. The first possibility was therefore ruled out. To distinguish between the second and third possibilities, Hilary Ellis performed lineage analyses. She basically followed parts of the development of N717 animals to determine whether the 131 cells that normally die were generated in the mutants, and if so, what their fates were. For this experiment, she had crossed the SED1 mutation E1735 out of the N717 mutant background. She therefore analyzed N717 in an otherwise wild-type background. And what she found was striking. The 131 cells were generated. However, they did not undergo any of the morphological changes typical for apoptotic cells. They looked like other cells, cells that are not programmed to die. Furthermore, the 131 undead cells persisted and survived throughout the animals' lives. We now know that many of these undead cells adopt a neuronal fate, which is the fate of their sisters. And not only do they adopt the neuronal fate, they often develop into fully functional neurons. These findings were significant for a number of reasons. First, the lineaging data revealed that what is defective in the N717 mutant animals is cell death, apoptosis. As I mentioned earlier, N717 is recessive and therefore most likely a loss of function mutation. The loss of the gene that is defined by N717 therefore causes a block in apoptosis. From this observation, one can conclude that this gene is normally required for apoptosis. Hilary Ellis had done the screen under the assumption that there might be genes required for apoptosis. At the time, in 1986, when the seminal work was published in the journal Cell, no such gene had been identified yet in any system. Their work proved that such genes exist, at least in C. elegans. Second, N717 mutant animals are alive, at least in the laboratory setting. This meant that a block in apoptosis in C. elegans is not detrimental to C. elegans development or to its viability. As we now know, this is not the case for most other multicellular animals. A block in apoptosis leads to a block in development in the case of all other animal models: the fruit fly, the zebrafish, the mouse, and of course also in the case of humans. The fact that C. elegans mutants with a block in apoptosis are alive has made the genetic dissection of this process in this model organism easier. Finally, the fact that Ellis and Horwitz could demonstrate that genes required for apoptosis exist and that animals mutant for these genes have a defect that is quite specific, a block in apoptosis, rather than very broad and pleiotropic, suggested that there is a genetic pathway that is specifically involved in and required for apoptosis in C. elegans and potentially also in higher animals. Now coming back to the second mutation that Hilary Ellis had identified, N718. Are N717 and N718 mutations in the same gene and therefore allelic, or are they mutations in two different genes? Basically, do they define one or two genes? How can you figure this out? You do something referred to as complementation test. This test can be done with recessive mutations, which N717 and N718 are. You take hermaphrodites homozygous for one of the mutations, let's say N717, and males homozygous for the other mutation, N718. You mate them and analyze the cross progeny. If N717 and N718 are mutations in the same gene, the F1 progeny will still be homozygous mutant for that locus, and therefore exhibit the mutant phenotype, in our case, the Z phenotype. However, if N717 and N718 are mutations in two different genes, the F1 progeny will be heterozygous for both loci because the parents were homozygous mutant for only one of the two loci and therefore contributed the wild-type allele for the second locus. The F1 progeny will therefore not exhibit a mutant phenotype. It turns out that N717 and N718 are different mutations in the same gene, which was named Z3, the third gene in C. elegans that can mutate to cause a defect in cell death, this time a general block in apoptotic cell death. Is the Z3 gene the only gene in C. elegans that can mutate to cause a block in apoptosis? Ellis and Horwitz continued to screen, however this time with an even better assay. To explain this assay, I need to tell you something about dominant mutations of the gene AGO1. And AGO stands for egg-laying defective. As the name implies, hermaphrodites carrying these mutations are unable to lay eggs. How do hermaphrodites lay eggs in the first place? On their ventral side in the mid-body region, hermaphrodites have a vulva, which is an opening through which the eggs are laid. The vulva opens periodically to release about 3 to 4 eggs or embryos an hour. The opening of the vulva is caused by the vulva muscles, and those are under the control of neurons. Most importantly, the HSNs or hermaphrodite-specific neurons, 2 serotonergic neurons. If the HSNs are not functional or are not present, the vulva muscles are not activated and the vulva cannot open to release eggs. The eggs therefore accumulate inside the animal. The HSNs are very interesting neurons because they are sexually dimorphic. They are sexually dimorphic because of apoptosis. The HSNs are generated in both hermaphrodites and males. While they survive in hermaphrodites, they are eliminated again by apoptosis during embryogenesis in males. Dominant mutations in the gene AGA1 cause the HSN neurons in hermaphrodites to inappropriately undergo apoptosis, resulting in adult hermaphrodites with no HSNs. These hermaphrodites therefore cannot properly lay eggs. Wild-type non-AGL animals have about 10 to 15 eggs inside their body. However, AGA1 animals accumulate 50 to 60 eggs inside their body, and this can easily be detected with a dissecting or stereo microscope. What this means is that to look at the AGL phenotype, animals don't have to be mounted on glass slides and viewed at a 1,000-fold magnification using a compound microscope equipped with DIC, as is the case for looking at apoptotic corpses in embryos or larvae. The AGL phenotype is therefore a very convenient assay for forward genetic screens. During their analysis of the SET3 mutations N717 and N718, Hilary Ellis had made a double mutant with one of the dominant AGA1 mutations. And what she found is that these double mutant animals, SET3 and 717, AGA1 and 1084, for example, were no longer egg-laying defective. They were no longer egg-laying defective because the HSN neurons survived again. This meant that a general block in apoptosis, as conferred by the loss of SET3 function, suppressed the AGL phenotype caused by AGA1 and 1084. That was great because what it meant is that the AGL phenotype of AGA1 and 1084 animals could be used as an easy readout for apoptosis. Apoptosis functional, AGL phenotype. Apoptosis defective, no AGL phenotype. And this is what they used for their second screen for mutants with a general defect in apoptosis. AGA1 and 1084 animals were mutagenized, and their F2 progeny screened for non-egg-laying defective animals, basically animals that could lay eggs again. One non-AGL F2 animal was identified that was homozygous for a recessive mutation, which was named N1162. In AGA1 and 1084, N1162 homozygous animals, the HSNs, as well as most of the 131 cells that normally die, survive. Is N1162 allelic to N717 and N718, and therefore another allele of the SET3 gene? Through complementation analysis, it was determined that N1162 is not a new allele of SET3. For this reason, N1162 defined a new gene, which was named SET4. SET4 represented a second C. elegans gene, which when mutated can cause a general block in apoptosis. Since SET3 and SET4 are required for many of the 131 cells that occur during C. elegans development, they are components of the commitment step, during which specific cells, the 131 cells, are irreversibly committed to the apoptotic fate.